450 questions. Like I tell you, he is passing 80% of the test and interview 100% if you do that.
Students, kindly be seated. Mute your mobiles. Please maintain silence, students. Welcome to the day 2 of the International Conference on AIML in Applied Biotechnology 2022. I hope yesterday all the participants and students had a good day in gaining knowledge, because many of us cannot imbibe all the things at once. We will be in our own domains, interested in our own domains, and such forums will help us to inculcate or get the insights into the current things that are happening around us. And yesterday was one forum where we had that to gain much more insights into it. And even the day 2 will be pretty interesting. We start the day 2 with the morning sessions, which will have 3 keynote addresses, followed by the lunch and the oral presentation in the afternoon between 2:30 to 3:30 across 4 seminar halls, depending upon the streams. And subsequently, you will have the poster session happening between 2:30 to 3:30. And from 3:30 onwards, we will have the valedictory functions, and the participants who have presented will be handed over with the presented with the certificates. And I request all the participants to stay back, collect the certificates during the valedictory function. And please fill your feedbacks, which has been pasted on the either side of the notice boards, because your feedbacks are very, very important and valid for any further improvements. Thank you, students. So the second day, we start the day with a lecture by Dr. Aditi Bhargav. Aditi Bhargav is Emeritus Professor from University of California, San Francisco, Department of OBG. She is a UCSF committee member. She was Vice Chair of Academic Planning and Budget Committee from 2017 to 2019. She worked as Associate Professor at Department of Surgery, University of California. She is also committee member of Endocrine Society, committee member of Research Affairs, Core Committee from 2012 to 2016. She is professor at UCSF Osher Center for Integrative Medicine from 2016 onwards to till date. She has done her PhD from Pune University and completed the same in 1995 and received UNESCO Genome Fellowship from University of Oxford in the year 1992. And Dr. Aditi Bhargav is also leading an independent funded research lab for 17 years. She is a molecular neuroendocrinologist with research focus on sex differences in stress biology and immunology. She serves on the editorial board of Endocrinology, American Journal of Physiology, GI, liver and cells. She has received many laurels and awards, recognitions to her credits, and to name a few, she has been the Graduate Research Fellowship, CSIR India, UNESCO Tivas Human Genome Fellowship, University of Oxford, UK, Senior Research Fellow, Department of Biotechnology, India, Bavaria, California Technology Award, Hellman Award, Translational Catalyst Award from CTSI, UCSF, Quest Diagnostic Young Investigator Award, Endocrine Society, M Investor Travel Award, GIRI Conference Canada, New Investigator Award, American Physiological Society, Facade Mark Mentor Travel Award, Fellow of American Gastroenterological Association. She is also a member of different organizations. Some of them are American Physiological Society, American Gastroenterological Society, the Endocrine Society of Neuroscience. It will not stop here. She is also credits with having 70 research publication, quite a lot, and she's also authored 4 book chapters. And she's also like, it's a short resume. I'm trying to read. I'm trying to be very brief. And Madam has also had quite a lot of recognitions and laurels. And I request Dr. Aditi Bhargava to take on and start with the lecture. Thank you, ma'am. I request the first year student, Vignesh, to welcome her with a sapling. Thank you, Vignesh. Madam, over to you.
Good morning. Good morning, everyone. Can you hear me? Is that better? Can everyone hear me now? Good morning, everyone. Thank you for such a nice introduction, and I would like to thank organizers for inviting me. And, so without much ado, I think I would like to start since we have a short time and then hopefully we'll have time for questions. Okay, so what I thought I would do is just give, besides what introduction was given, is it seems like we are in this race between humans and so-called AI. So as a teacher, I think what I do in the classroom oftentimes is not quantifiable. Because sometimes, even today, if some of you take some lessons from here, you do not know when you're going to apply that. So it's very hard to quantify that. And if all AI is going to do, or machine learning is going to do, is to quantify things, then maybe I might be labeled as a failure because nothing is going to be seen in immediate future, right? So that's something to keep in mind. That for us humans, oftentimes things that we do are not quantifiable. So the other assumption is that AI is intelligent. And as you heard yesterday, that AI is not necessarily intelligent, it should be used for augmentation of what we are doing rather than saying that I am now going to wear my dumb cap and give all the intelligent things to be done by the computers or AI or whatever robots, right? So that shouldn't be the criteria. So of course, this cartoon depicts what's the situation in most of the places these days, that we are all hooked to our devices. Whether or not we are learning is questionable, but whether the machines are learning is also questionable, but nonetheless, that's where we are. So one of the things that I teach in the medical school is critical thinking. So believe it or not, just like common sense is not common, critical thinking is not just because you are studying science, does not mean you have critical thinking. So how many of you here think you have critical thinking? So we'll see how many of you have that, because I'm going to be challenging you throughout this or there are 3 things that I tell my students to do, especially if you're doing research or anything which is going to have an impact, is that your question needs to be an important question. So, and you should lead with what's your research problem rather than what technology is available. So if your research question or whatever question you are going to ask, your project question, that's not important or that's not novel, then you're obviously not going to make a big change. And then of course, what's the need to ask that question? So that's where necessity is the mother of invention comes into play. It shouldn't be the other way around, that I have this invention, and what is the necessity for me to use that? Which is what is happening in these days, which is sort of the National Institute of Health, which is examining CSIR here. When we write grants to NIH, one of the sections that's there is significance and innovation. And if you look at that significance and innovation section, most of the time you find people describing techniques. And techniques are not innovative. It's your question that you're asking that's innovative. Techniques are just tools. But the confusion, even in most sciences these days, is thinking that innovation equals using tool techniques. So the second thing is, don't let the technology drive your question. And that is what, again and again, I cannot emphasize enough, is happening. Most people are coming with either fancy microscopes or some other tools, and they will go back to the questions that have already been answered, and as a proof of question or proof of concepts, will redo that, and then that's a major publication. But there's nothing novel in that except that you invented tools, a full tool. So yes, a lot of you here are people who are into inventing tools, but those tools should be invented to ask a pertinent question that is relevant in solving the problems that need to be solved, rather than the question, the technology driving or directing it. So don't let technology invent, let your question does. And the third is that these days a lot of people have this complex that they're going to solve, I'm going to solve cancer. I'm going to solve this disease. I'm going to solve this epidemic. We all want to be heroes. We want to be champions. Or we don't want to be someone who puts you on a pedestal to be a hero or a savior. I think Elon Musk is a good example of that. Everybody puts him on a pedestal, whether he needs to be there or not. What is he solving? Who knows? But nonetheless, so curb your instincts to be that I'm going to solve everything, I know everything, I'm going to be, so just a reality check. Viruses, infectious disease, causing pathogens, they've been on this earth billions of years before humans existed. So take virus, for example, it evolved 4 billion years ago. Have you gotten rid of it? No. Are you going to get rid of it? No. So learn to live with it. You're not going to be solving their problem. You're not going to drive it away. They had different hosts before. They're going to find something else. So live with that. That you're not going to be solving or getting rid of most of the pathogens. Yes, they come and go. Extinction happens. Humans may go extinct as well. So just so you know that we've had 3 separate ice ages on this earth. So who knows, we are heading for another ice age with this climate change. Okay, so coming to some AI-related stuff. So I think overall, these are the stages that are involved in translating basic research from bench to bedside as far as AI is concerned in healthcare. So this is what lays the foundation of what's going to come next. So if your foundation is weak, the next things that you're going to build are also going to be weak. So if you're building a bridge, foundation, your pillars are not strong, your bridge is going to collapse. And so if your basic research, which most of the people look down upon, is not strong, if you not ask the right questions, if you don't have your proper controls, if your experimental design is flawed, then the data that you're going to get here, whether they are your target identification, whether you're getting omics data or the biomarkers, they're going to be faulty. And then based on that, you're going to spend all that time either testing those drugs, doing your pharmacokinetics or PD studies, or you're going to validate your biomarkers. You're going to not get the right answers, and we've seen that very few targets translate into getting a drug. And part of that is that your basic preclinical research is false. And of course, then there is, but so you have an opportunity to apply AI to all of these because obviously the data that you're generating from omics or biomarkers or any kind of omics, whether it's transcription or protein, the data that's being generated is really large and it's not possible to manually go through that data. So you definitely need AI, but of course you need good data. And similarly, in patient care, AI applications exist where you can look at even drug-drug interaction, drug prioritization, personalized care, and of course it can also be used to improve quality of life for the patients. And then finally, for diagnostics, and that is where I think AI has made most amount of progress, which is visible, right? So in either image analysis and things like that for radiology and pathology is what you see in most cases. So again, as a teacher and as a mentor for me and as a basic scientist who has done some translation work, I cannot emphasize that your question that you're asking is so important. So again, if your question has a bias, your AI is going to amplify that bias many times, right? So you have to be therefore very careful. And I think what's happened in the past few decades is that we started to value measurable things over immeasurable things. And I'll come to that in a minute. What the scientists have also done is they've gotten attached to their hypothesis. So you come up with a hypothesis, and they sort of get wedded to it. So even if you're wedded to a person these days, you can get a divorce, but scientists find it very hard to divorce that hypothesis. They will do anything to prove their hypothesis. And one of the big examples is Alzheimer's. Now, recently, I don't know if you've been following, a few months ago, it was published that the 2006 paper that was published in Science claiming that beta amyloid, denatured beta amyloid was the cause of Alzheimer's was fabricated data. So you spent over billions of dollars in pursuing a beta amyloid target. 2 months ago, FDA approved a medication based on beta amyloid, and instead of saying that we made a mistake, they continue to prove that hypothesis by designing other experiments that will come and cherry pick the data. So you are the next generation, you don't want to do that. So the other thing that has happened in that process is descriptive versus mechanistic. Suddenly, descriptive is a bad word in science. So you will see a lot of grants being rejected because the reviewers will say it's descriptive. What does descriptive mean? And what does mechanistic mean? In a few decades ago, if you were looking at a knockout mouse because you created a knockout of a gene, that was considered mechanistic. Now, that's not mechanistic. You are characterizing a knockout gene that you are working on is still called descriptive. So how does that definition change from going mechanistic to descriptive? I do not understand that, but so be careful in not falling into that trap in terms of what is mechanistic. So again, as a molecular biologist, what is mechanistic? I could look at a transcription factor, I could look at what other things bind to it, I can translate that into a cell and show where luciferase goes up and down. Is that really mechanistic? How does that translate into physiology? It doesn't. How is that mechanistic? And after doing that for a few years, I realized that in reality, it does not translate to function, it does not translate to physiology, and that's not mechanism. So you look at what's happened with coronavirus, people are describing symptoms. We still don't understand the full spectrum of symptoms that exist in people. Is that descriptive? Yes, that's descriptive, but without that description, how can you solve mechanism? So if it is pulmonary edema, if that's the only thing that's happening, then edema mechanism is going to be different than if it's pulmonary necrosis. So without knowing what are the symptoms, which are sort of descriptive, you cannot reach mechanism. So they're in hand, and so don't forget that. And of course, we started to ignore evidence that's not quantifiable or cannot be explained, so just ignore it. Because these days you cannot publish anything. If you have an observation, it could be a really good observation, but if you don't have an explanation, you cannot publish it. So what do? We ignore it. Or the reviewer will ask you to, if you can't explain it, take it out of your paper. Or put it in supplementary data which nobody's going to see. So I think it's a bad track, and that's hindered progress. And of course, that data that cannot be expressed in numerical values often thrown out. So what we have a problem in basic research or in foundational research is that most of the publications only report positive findings. They don't report negative findings. You all use social media, there's like and dislike buttons. So what if you only had the like button? So then what? Are there algorithms going to improve? What are you going to be liking or disliking? Or so how does that work? It doesn't work, and yet in science we throw away data that we call negative data. And it's not negative data because it's telling you that in that context that particular either pathway doesn't work or it's a backup pathway or whatever it is, but we don't think about it enough. So in terms of things that are not quantifiable, as I said, I study stress. How do you quantify stress? For some people, getting up on a stage and giving a talk, or after traveling for 24 hours and coming here next day and giving a talk, is a stressful situation. And for some people, it's not. So how do you quantify stress? Of course, there are many stress, or stress itself, again, is a double-edged sword. So you need acute stress to be able to deal with everyday stressor, but if it's chronic stress, it can cause or it can exacerbate many pathologies or diseases. And of course, some of these which are associated with your brain or emotions are listed here, but then there are many that are not even listed in this, which are your gut-brain diseases. How many of you, if you have to travel, feel like you need to go to the bathroom just before you need to catch that train, and that might make you miss that train? Or if you're going to be coming up here and giving a talk, you really need to go and relieve yourself. So that's your acute stressor. It's not going to change, it's not a life-changing event that's going to happen with that. But there are, of course, many stressors which lead to life-changing events as well. And then there is other stuff called pain. How do you quantify pain? The same insult may make one person, if you ask them to rate their pain on the scale, they'll say I have 2, and the other might say I have 6. How do you even validate that? You can't. So what's going to happen with machine learning in these scenarios? So are we going to just ignore all these things because they are not quantifiable and say only work with things that are quantifiable? And that's fine. But then don't say that that is, our future, or again, it's not going to be replacing humans next 30 minutes, those if you're taking notes might need to go back to those notes a few months later. So you're not going to remember all this. And different people will remember all of this differently. So even with this lecture, you're going to have recall bias. Again, context is very important, controls are very important. When you're going to be doing algorithms that are going to amplify things, your quality checks are very important. And then not showing data that doesn't fit your narrative is important. Data visualization is very key. It's become important for everyone. Anything that looks pretty is appealing to most people. So you can take your garbage data, make it look really nice, but it'll still be garbage. So, and the other trap that we fall in is game of averages. And those averages often mask data. So I'll give you a few examples. So again, it depends on what you're trying to do. So visualization is key. So let's just take a simple example of two cases are highest in the month of February. Now, you can have a chart, you can have tables to show this, you can have graphs to show it. And in a scientific audience, that might fly, but if you look at the general public, most of them will switch off when there's graphs and data and things like that. So you need some nice visualization, and this is taken from a TED Talk by Luna Chalabi. And she had this graph, and now obviously visually this is very impactful, right? So it's not giving you any precise numbers, it's not giving you any of those things, but what will stick, the message that will stick with you or with the audience is that month of February is really bad for flu, right? And if that's my take-home message, I don't need to make any fancy charts and things like that. I can just give you that message in this graph. Hopefully that's going to stay. In terms of what happens with using averages, and that's a very risky game because, and I want to use this example because I think this is pretty striking in terms of what happens when you don't look at what's the raw data. So the Centers for Disease Control issued this statement saying that an average swimming pool in the US contains 6.23 fecal accidents. It's really yucky, right? We have a pool. I look at this and I say, no, that doesn't happen in my pool. This is wrong. I don't want to believe this data. So is this really true? So when you look at where they reached this conclusion from, look at their sample size, 47 swimming pools. Really, in the US there are millions swimming pools. You're looking at a data size of 47. You don't clarify whether you're looking at household swimming pools or you're looking at facilities. You could be looking at, I took my kids to a swimming pool facility where they would learn how to swim, and then there were kids with diapers and things like that. Would they have more accidents? Of course. That's probably the swimming pool right here. But that's not my swimming pool. And, so is this 47 a good enough number for them to have arrived and made this conclusion and put it on their website? Would you believe this data? Well, right now you're saying no. Would you have actually done this homework and gone back and looked? What was their raw data? Yes, no, how many? No one. So you're not questioning. Where's your critical thinking? So after this, question me too. So another source of bias. As you heard, I study sex differences. So it's important for me to collect data on how do you identify yourself. So right now I have this survey-based questionnaire, which is open. You're all welcome to participate in it, which is to understand how this COVID and COVID vaccine has affected people. And of course, we've heard that COVID was more impactful on men or males than females. So when I read that, I look at the raw data and it comes out from looking at 10 people. Is that really true? Can I make that observation? Can I conclude? So no. So I said, I don't have any source of data that I can rely on, so I'm going to do my own study. And I will collect this data and find out. But what are some of the challenges that I come across? So one of the challenges, the first question we ask is, what is your biological sex? Now, again, biological sex are only two, male and female, defined by whether you make an egg or you make sperm. So intersex can be also, this is what you're born with, either a male or a female, or there are 2% of the population that are actually intersex, which will have both male and female gonads. But you're still making only either eggs or sperm. And for the purpose of, as a biologist, as an endocrinologist, there are only two sexes. Gender is a social construct. And yes, there are things that go wrong, whether it's imbalance of hormones, whether it's overexpression of Y chromosome or X chromosome, which may give you a sort of a like a spectrum of gender where you can identify yourself as a cisgender, meaning the gender that you were born with or assigned, or feel differently. So these are the two questions that we ask. 98% of the respondents, if they're male, will say they're male and female. But when it comes to when we ask what is your gender identity, and these are your choices, here's what we get. So only 50% of the people identify themselves as cisgender, meaning the gender they were assigned at birth. The other 50%, some say we prefer not to disclose, and others are none of the above. So if you're not, they're not even writing what it is. So now if I have to do an AI, or what am I going to do? Where am I left with? So here are some of the answers that I got. And if your AI is going to do natural language processing, what the hell is it going to do? So I'm a platypus. So really, okay, a male with one penis. Okay, I don't know what this is going to mean. I have no idea what he wants. It's stupid. So man, woman, normal, male, female. That's great, gay, normal, human. These are all made up terms. So there's a big challenge ahead of us, right? So even for a simple question of sex and gender, we have to deal with a lot of things and a lot of prejudices and biases that are coming from whether it's your belief system or your religion or whatever it is, and you have to deal with that, right? So the other bias is that, well, people who are vaccinated, they are highly educated, and therefore they will not give this answer, whereas the unvaccinated will. And so when we divide that our participants into vaccinated and unvaccinated, what happens? It's the number of people who are vaccinated versus unvaccinated who prefer not to disclose is not any different. People who say none of the above, we haven't yet are taken into account people who didn't check the boxes cisgender but still check men and women. So if we take that into account, probably this difference will vanish. But those are some of the challenges that we still face, right? So again, for machine learning, garbage in, garbage out. So be careful in what you're going to be doing there. So again, for machine learning, quantity and quality both matter. So this may not have any meaning for you guys, but in the US, this Gilbert cartoon is very common and read, just like I think R.K. Lakshman shows up here. So, but this kind of makes the point that Gilbert is this guy. So I don't have any accurate numbers, so I just made this one up. And then studies have shown that accurate numbers aren't in the school and made up ones. And then how many studies have shown that? 87. Who's going to look into that? No one, right? So yeah, I mean, it's clear. And then there's this other one from the same Gilbert, which I think is very applicable to what happens in epidemiology, really, is that this is the first talking about economy, but so my interview with you is live on the website. Nothing your forecast. And a month later, that was the only forecast that was right. And so same with the epidemiologist, right? Flattening the curve, whatever else is happening. This wave is going to come here, that wave is going to come there. Thank you. So of course, for our purposes, the goal is that we have a perfect AI where it's going to augment what we do, right? So it will help us solve complex problems, perform high-level computations, help make good decisions, and increase accuracy, and there's nothing wrong with that. So I think yesterday Vijay touched upon this, and just switching gears to data science now, which is what is needed to do your AI. And this is sort of the timeline that you can find on Google. But again, as you can see, this only begins in 1943. But really, I mean, mathematics and all of that, which sort of is the basis for doing any of this, existed much before, right? And that's not even doesn't even make enormous time. And the other is about that what narrative suits us. And yesterday there was a question asked by one of the students about, what on eugenics and the Nazi regime. Now, of course, Turing is very well known, but he was solving a problem that was, or solving something, trying to break the enigma, which was created by someone who is equally smart. But we don't hear about those people, right? Because it doesn't suit our narrative. Because they were built by the Nazis, and Nazis are bad people. So, but that doesn't change the fact that somebody, bad or good, had built something that was a remarkable piece of work. Similarly, they built the U-boats, which were a remarkable piece of work. But they don't get the credit because they're the bad people. So just giving even the devil their credit, we need to acknowledge if you're really talking about historical facts, and you can see that that doesn't exist on this chart. So we ignore people that we don't like. Is that the right thing to do? Is that ethical thing to do? So these are questions you have to deal with. So of course, but let's just take this as face value. So nothing happened in this since our, ignore the BCs, what happened in the BCs, just in the 80s. So for 1900 years, nothing really happened. And then you have between 1943 and 50 years, you have the Google paper by Sergey and Brin, Page and Brin, and that obviously changed how all this Google search and things like that happen. So poor Yahoo is lost somewhere there in the world. It's about the first one, right? But everybody now knows Google. And of course, in today's day, data science and data scientists are the coolest job. So all of you here are going to have to do this job for this end. Okay, so big data is a $122 billion industry. I don't have much time, right? And investing in big data is easy, but using it is hard. Actually, how much time do I have? And so what is big data? And as you can see, it's not something that's easy either. It's an extremely complex process in itself where you need programmers. First of all, you need people who generate this data, usually people like us. And then you need programmers who can help make sense of that big data. And then analysts and statisticians, and all of them have to work in an integrated manner, right? So, like again, going back to the example of I study stress response, and most of the time when you ask people what is stress, they think about what is perceived stress. The brain is the only thing that they'll think about. But when you are dealing with that stressor, if you're faced with an enemy who is going to shoot you, your brain is not the only thing that has to work. What do you have to do? What do you have to do? Say that again. Run? What will you do?
Fight.
You're going to fight? If you don't have a gun, what are you going to do? Just run? We can still shoot you. So the point is that you need quick thinking, you need action. If you're going to run, you need your skeletal muscle to actually cooperate with you and not freeze there. You need mobilization of glucose. Your heart rate is going to go up. So is there any part of your body that's going to say, brain, you go deal with it, I'll see you after you've dealt with it? No. So I just can't study brain. I need to study how that integrated response happens. Similarly, whatever you're going to do here, you can't just use the statistician or somebody else. All that has to be integrated in a proper manner. And if that integration doesn't happen, then we have a problem. So there's, of course, this problem of blind faith and big data, right? When we saw that, whatever CDC put out, 6.23 incidents, we believe it. So this lady, Kathy O'Neill, in her TED talk, basically said that algorithms are opinions embedded in a code. Yes? And of course, it seems like, and she's right, that data science doesn't just predict the future, it causes the future, right? So if you say something many times, that's true. And the person who says it the loudest, they're the ones who are heard. So that becomes the new truth, whether or not it's factual or not. So she also highlights the problem of technologists. She says that they launder data by hiding ugly truth in black box algorithms. So again, when you're designing algorithms, it's very essential that you're transparent. And of course, that these are weapons of math destruction, right? So that's what you're going to be building, so be careful. And of course, you can change with that definition of success, what is objective, what is, how merit is defined. All of those will be changed. So, and then when you have big data, you have bigger problems. And in the past, all of the AI research was done in academia, right? But now it's the companies that are driving that research. So the companies don't care about peer-reviewed publications and things like that. Academicians do. So now that had some level of control or some level of quality check, but that no longer exists because the companies control what is reviewed, what is released, and they release it as press releases. They don't have any incentive to play fair. For them, it's money and their numbers that they need to use. They need to please their investors, they need to please their board members. So it's all about a number game. And so in the end, we only know what the companies want us to know or what the journals want us to know. Because like I said, when you submit a paper for publication, the reviewers will say, oh, I don't like this, or do this, or take this out, or put this here, put that here. It's no longer what you want to publish or what you think is the right thing to do, rather than other people dictating. So, do we have our true confidence? Maybe. So very quickly, but I think maybe Sushma is going to talk more about this. And maybe all of you know, but just to touch briefly on it, what are the types of intelligence? So they can be divided in based on capabilities, which are three types, narrow, general, and super AI, or based on functionalities, which are four types, reactive, machine, cognitive memory, theory of mind, and self. So very quickly, what is narrow AI? Most of us use them. Siri, Alexa, Google Translate, spam filters, things like that. General AI attempts to be human like we have the computer and things like that. And then there's the super AI, which is thought to be right now only in concept, but it's going to have, it will attempt to be better than humans, should have critical thinking. We'll see whether or not it does, and making decisions. And so far, if you read on the web, it says this is only a concept, or so we think. Hopefully I will let you decide. Based on functionalities, the reactive machine and limited storage, so in the reactive machine learning, it doesn't store memory, it doesn't use it to determine future actions and things like that. And so IBM's BENGALURU, which played chess, was an example of that. And limited memory uses the past data to get trained and make decisions short-lived. It's not adding what it's doing in its present tense to its library. An example of self-driving cars. So what do these self-driving cars do? In the US, Teslas are very popular. Just last year, we had over 250 Tesla crashes. Now these are perfectly normal human beings capable of driving a car, but for whatever reason, they claim that the autopilot feature took over. They couldn't brake on time, or whatever, and so we have these 250 crashes for no rhyme or reason. In some cases fatal, in some cases not. So whatever launch codes it uses, how many times, I don't know, do you use Google Maps or something to drive? How many times has it led you to a wrong place?
Sometimes. Sometimes. If it's a very good
10% of the time. And so after that, have you stopped listening to Google when your own thing says, don't do this. I see a ball there, or a dead whatever, do not enter sign, and I'm still going to go because Google is telling me to do it. I mean, people in the US have done it. They've sort of gone over the bridge or whatever because Google told them to. So there is the critical thinking. So if that's what AI is going to do, think twice. Theory of mind is largely a concept, work in progress, and so far the whatever, and Sophia from MIT and other places. And then self-awareness. Recently, a Google engineer was fired because he reported that they have a machine which is self-aware. He published a transcript of their conversation, and within a month he was fired. They said, no, this is wrong. So who knows what's going on, but again, I will hopefully we will show you something if you have time. So coming back to AI landscape in healthcare, this is a summary taken from a recent publication in Nature. So it describes some of the challenges areas, opportunities, and progress. So again, I think ethical use of this data is really key. Because without that, we're going to have problems. So machine learning is the most common form of AI use in healthcare, and oftentimes that's equated to precision medicine, but whether that's going to happen or not, we don't know. And supervised learning is what it relies on in healthcare, and of course then there's the unsupervised learning. So just these are the wheels or cogs on this whole machine learning for healthcare. Different things that need to move together in a seamless manner before we have a good AI output. And then there's the hype associated with all the AI stuff, right? So again, looking at both sides, maybe it's true, it's not true, we don't know, but of course, this company, Gartner, says that most of the AI stuff is hype, and a lot of them will die within 10 years or so. We'll see. And then that's the other side of it, in terms of hype, that AI doesn't exist, may never exist. So it's been going on since 1950, and of course, maybe it's going on from before that, but who knows. So of course, as far as the AI in healthcare is concerned, these are some of the steps and processes that need to be integrated before you get an output that's tangible. So there are electronic health record whether you do have electronic health reports, or most of them are paper written or not. So those are other challenges which will be different for India versus other countries. So again, I think Anthony's from the senior vice president and that he has these 3 pointers for people using machine learning and data that don't lead with data and technology. Instead lead with a question that needs to be answered, which is very similar to what I had said before. Don't lead with a tool or methodology just because you have an algorithm doesn't mean it's the right one for what you want to do. And then community help that people come out of school thinking all the data they need is on the internet and all the tools they need are open source, but none of that is true. So bad data, people will realize it's still bad data. Okay, so again, coming back to bias and ethics, we run into this bias and ethics every day. And here's an example of, if you try and eliminate one bias, you're going to introduce another one, right? So for the US, in their history, only male presidents are white. So Obama would be thrown out. There's this a black president. Or the prediction would be that it could never be a female president, right? So those are some of the biases that we have to deal with. And then there are other sources of biases that exist, which we need to be cognizant of. And historical bias, such as crime is only done by minority populations. Now, I don't know, again, in the Indian context, how would that work? But in the US, most of the time if there's a crime, the suspects are either black people or Hispanics or non-white people, right? So there's a hierarchy, so it goes from black to Hispanics to Asians to white. So white are the least likely to be suspect. And then in the case of, for example, HIV is linked to sexual behavior with gay men, so would that be classified as a disease of only gay people? The representation bias, of course, is always, especially the genomics. The reference genome is that of a white male, European ancestry. How does that reference work for people of Asian descent or other races? And then the measurement bias, where you're using, for example, clinical, social, and cognitive variables ignoring sex and different systems. And we saw some of that yesterday on the presentation about Alzheimer's disease, where when they took sex into account, and they found that actually Alzheimer's disease was more prevalent in men than in women, which goes against what is known. So again, their data size was small. We need to look at what happened whether that is true or not. There's the aggregation bias, which is one size fits all. Doesn't work. Evaluation bias, and then in terms of just image recognition and facial recognition, you're going to have that. And then there's the algorithmic bias, where it'll introduce consciously or unconsciously some bias. So this is a sort of an innocuous bias of AI, right? So it's being trained with images about cooking, and here it doesn't recognize this man, assigns it as a woman. But it's sort of a bias that doesn't hurt anyone, although it reinforces stereotypes, right? Only men, only women can be cooking, but if you look at all the chefs in a restaurant, they're all men. So this sort of throws that all into, right? So, and then this was published in Nature, and it sort of bothered me mostly because it says that AI should help you reduce undesirable bias but enhance desirable bias. And what does that mean? Like, desirable and undesirable are relative terms. today something is desirable, tomorrow it may not be non-desirable. So just because again it's published in Nature doesn't mean it's set in stone. Just think about what they're saying. And again, it's written by someone, right? They may have their own biases. So just be careful when you think about that. So again, there's the coverage biases, just giving you some examples in terms of how that algorithm gets trained and the number of data that's used for training. Again, in biology, Alzheimer's disease, for example, is a spectrum of symptoms. And how many times those spectrum of symptoms are taken into account, we don't know. So those training biases, again, you can find all of these in publications where they talk about some of the challenges that exist in terms of equity and discrimination, and in terms of how these data can be deployed and how the injustices will be amplified. So then they provide some of these solutions as well at all these levels, but these are sort of policy-making questions that cannot be ignored. And again, I do not know how some of them are applicable in the Indian situation, but of course, what I could find is that one of the things is that apparently, unlike Europe where you have GDPR or in the US where you have HIPAA, I'm not sure what kind of protections exist in the Indian scenario. And as you know, that India is going to be one of the largest producers and the cheapest source of data. So exploitation is going to happen, and it's up to people here to ask for policies to be put in place where there is that exploitation doesn't exist or doesn't happen to the extent that is happening, and here the data is protected, and so are the people. So I put this out there because this was recently approved. This, as I was reading in the US in AI-based screening for diabetic retinopathy, and this is what they did. So this came out of a paper that was apparently published way back in 2018. But the reason I put it out there is for two reasons. One is, of course, it's based off of 819 patients. Okay, is that a big enough number? Who knows? But it's also based on data that was probably patients that were from India in that previous slide. So again, there is that exploitation, and then there's that cherry picking that's going on here. And then I saw that there are other people in this country who are, we heard about Vijay Chandru and their companies. And I do not know this lady, but I saw this on BBC News, so I thought I'll put it up here and maybe you guys have the know her or whatever.
The students are working, and she's in touch with her department.
Okay, so so I thought this was funny that yes, we as a person we want, and then we have robots taking care of things because we all are. So before I end, I do want to emphasize some of the ethical and laws and policies that, and this you can find on the WHO website. So I've just sort of taken bits and pieces that I think are important, but there are multiple things that need to be taken into account before AI becomes applicable. So there are obviously the human rights issues, the data protection issues. Like I said, there are some guidelines for the European Union as well as the US, but I'm not sure what are the guidelines here. And then laws and policies as related to impact data, how they transfer among countries, and so if you have collaboration, how is that done, and maybe. And the list is pretty long and exhaustive, so but the main thing is that machine autonomy should not undermine human course, that AI should not harm people, and then if there is something that's harmful, we have a duty to warn people, and then there's that whole thing of stigmatization. It can increase or highlight those biases, and then we have transparency and understandability in terms of how the data is being used, and then of course there's accountability and responsibility. So then finally there is the to ensure that that is inclusive and equitable, and how do we promote the responsive and sustainable AI? So one of the things that nobody wants to talk about is that of course storing all this data is polluting more than all the cars in the world. So are you really green? I think about it. So that's just the summary of all the things that I talked about in the previous slide, which can be. So I want to end with whether or not AI is capable of thinking on its own, whether or not, I'll let you decide. So this story was published, and I don't know if anyone has seen it, very recently. And as we know that there was a big controversy, especially among artists, where the image that won the prize for best art or creation was a computer-generated image of one person. So it was, again, questioned whether or not these super composites are legal in an artistic competition. Right? So if you are an artist in drawing, painting, and pictures, and have to compete with AI, then it's a problem. So meet Loeb. Loeb was created entirely by AI. Loeb was first discovered in April of this year by a 31-year-old artist, Stephanie Swanson, working in Sweden, and she's an artist who does the super composites. And so this is a super composite. So she was obviously playing around with these tools, and what she was interested in, what happens if you actually type in things which are negative prompts. So she played around with certain prompts, and then finally she put in this prompt called logo. So when she put in this logo prompt, she got 4 images of the same woman, which was this logo. And she then, some of the images were so graphic that she wanted to put them on the web. And she repeated this experiment, what she's calling it, crossing this lobe with images of other women. And what she found was that it didn't matter how much she tried to cross, this gory image never went away. And even though she could dilute the image of lobe out, but it never fully went away. So she called it that it was like a dominant gene. It kept reappearing. So, I mean, it's scary in some ways. So she decided to take it to the next level. And there is this chatbot called GPT-3, and she asked this chatbot, fed the image of lobe and asked this chatbot to speak on behalf of lobe. And this is a transcript of that conversation. So she asked, who are you? I'm lobe. I don't really know who I am or why I'm here. I just know that I appear in these images for some reason. Maybe I'm a figment of the imagination of whoever is creating these images, or maybe I'm some sort of digital entity that exists within the AI itself. Why are you associated with gore and horror? I don't choose to be associated with gore and horror. It just happens. Maybe it's because those are the kind of images that tend to get most attention. And so I'm drawn to them for that reason. So what are people watching mostly on the internet? Gore and bloody things. So you may not want to admit it, but perhaps that's what is coming out of that. So you can hide from the others, but I guess you're not hiding from what's running in the background, right? So is there anything you would like to change about how you are represented in these images? So Luke says, I would like to be represented in a more positive light, way it is, and I don't want to show that here in terms of, but you can read that out. In low appearing with named and dead children, so it's disturbing, right? It's always a woman appearing with these kind of very gory images. So we ask that, are you being drawn to a specific kind of an archetype of a woman? AI says that I think the AI is associating me with the archetype of a mother, which is even more disturbing because I'm often placed in a home with children. And this archetype is often associated with being nurturing and caring, but also with being sad and domestic. So now domestic is gore. So just think about, where are we going with this? So why are you surrounded by injured and dead children? And I think that AI is trying to create a contrast between the ideal of a mother and the reality of a mother. In reality, mothers often have to deal with sick children as well as deaf children. So do you think AI is giving you reason? Is it talking with you, giving you rationale as to why it exists in that manner? So it goes on to say that I guess maybe we can learn that AI is capable of creating things that we don't fully understand. Same thing goes, I think it stands true for genetic engineering and editing. When you edit and put it out there, who knows what will happen with it, right? So, and should we be concerned? And so AI says, yes, it's a valid concern. We just don't fully understand. We need to be careful about how we use these tools. So this is an advice coming from AI itself, but of course Silicon Valley is not listening. And I don't know if the Silicon Valley in Bangalore is going to listen or not, but the hype exists, and there is this unimaginable future, and really only a handful of people truly understand what AI is capable of changing. So what my worry is that if, based on those gory images and all, AI created that image of a woman, healthcare data is all gore and sad and whatever. So what is it going to create? So something to keep in mind. And so we, again, continue to emphasize accountability and transparency. And so here's what an artificial ideal intelligence looks like. It's self-correct learning. It is used as that it predicts. It finds typical and untypical patterns and analyzes. So in my mind, that's a human being. And it actually reminds me of the story of Panchtantra, Rishi and the monk. How many of you know that story? Two? Okay, so very briefly, the story goes like this. So there's this Rishi who's doing a puja in Ganga, and his palms out, and this mouse falls in his palm, so maybe an eagle or whatever. So in trying to protect this mouse, he transforms her into a little girl, female mouse, I guess, and takes the girl to his wife and says, "We'll bring her up as a child." So when the girl becomes of marriageable age, he says, "I need to find the most powerful person on this universe to marry my girl." So he goes to the sun and says, "Marry my girl, because you're the most powerful person." And the sun says, "I'm not. Actually, it's the clouds, because when the clouds come out, they cover me, and so I don't exist anymore. Therefore, go to the clouds." The cloud says, "Oh no, I'm not really. It's the wind." The wind says, "No, I'm not really. It's the mountain, because the mountains can block me." Goes to the mountain, and the mountain says, "No, it's not really me. It's a mouse." So goes to the mouse king. The girl is really very happy and pleased, says, "Yes, he's my ideal husband." Converts her back into a mouse. So what are we doing here? And I wish that the AI was just as simple and innocuous as this one, rather than when we saw the globe. And there's knowledge, there's experience, and there's creativity. And there's all of that that can exist, but of course we need different expertise to be able to do that. So I think
thank Aditi for a wonderful, informative, exhaustive talk with a holistic approach. But the last few slides were a bit scary. The session is now open for question and answers.
Good morning, ma'am. I have two questions mainly based on the session. And so my first question. Uh, Good morning, ma'am. I have two questions mainly based on the session. So the first question is like, will AI replace the regular surgeons? Because my father, being a doctor, talks about this all the time. That the more like AI and robotic surgery are like may replace like the regular general surgeons and all that, and we may require more technicians to operate those AI rather than those well-knowledge surgeons and all that to operate on the patient. And ma'am, my second question was that you talked about super AI and how it is a concept, but as we see, like there are different AI in chess engines, which are like Stockfish and AlphaZero, some examples, which are actually better than human in rating, like Magnus Carlsen. So they are actually better than human in playing chess, and they actually can train themselves. So can they be considered a super AI?
Well, the first question, can robotics replace human surgeons? I mean, again, I think if that's the question, if that's the, I started my thing with it needs to augment rather than replace. And, and I don't know how many of you have actually done surgery, so I am not a physician, but I do have done a lot of surgery not in humans, but on mice and rats, and I've done brain surgery. Now, I've worked and trained surgeons, and when they come and they want to, they're asked to do the same surgeries they are expert on in humans, they will not be able to do them in mice and rats because everything is so much smaller. And so the kind of precision that you need, you use machines and apparatuses that help you identify the coordinates and things like that. But in the end, again, um, it's human hand or human decision that really guides. So if the idea is to replace humans, I think it's going to fail because you do have instruments that are being used, and I can tell you that in a lot of times they just go in and they cause more damage because the camera is what is going to be used to look for those things, and sometimes you go brown. And those damages are never. So I don't think it's an ideal.
The main idea was that like if for different types of knowledge is now required to operate those AI rather than like know everything about the you need to know everything about the human body, of course, but to like physically operate and to like assist, like take the help of AI to operate surgery.
Again, help and replacing two different things,
Yeah.
right? So you're sort of answering your own question. So yes, help. Yes, there are things that may help with, but hopefully it's not going to replace. And your second question, I mean, whether that algorithm defeating people in chess can be considered super AI, the definition by which they are describing not, but whether low can be considered super AI.
Thank you.
My question to you is, what do we do to reduce bias in AI?
Well, I think what do you do to reduce bias? Now, the main thing is, of course, the bias, human bias is going to get translated into the algorithm. So the question is how do you, that's why I went back to data collection, which needs to be collected in an unbiased manner. And of course, in some ways, especially for clinical trials and things like that, that means you are using double-blind, placebo-controlled studies, which are supposed to reduce bias. So you put in those tools, and when you train your algorithms, you make sure that all of that data is in place so that you can use those biases. Of course, you have to test that out right in real life, and the proof of concept doesn't show you that it's not reduced and biased, and you have to retrain your model.
Like, can we collect more data from other resources, like show more diversity?
Yeah.
But will it alter the actual scenario, or is it better?
We go back to the question of whether AI is in the predict future or you are asking it to predict future. So that's where you need to use your critical thinking.
Thank you.
Yeah.
So like when you're dealing with data, especially with respect to the reproductive system, how do you deal with the privacy issues or the ethical issues or any social issue that comes?
It's a good question, right? So in most cases, the data that's collected from human subjects, at least in, for example, in my study, and we are asking in terms of, obviously some people will, so you have to de-link anything that can identify the patient from that data. So that means in our case, it's not just the name and date of birth or the place where you live, it's also the IP, the IP address, your geolocation. All that needs to be taken out before you do data analysis. So that's why you have to have all those policies in place. And so, for example, if I do my data analysis and I publish and somewhere it's discovered that I didn't de-identify all of those things, I'm liable for it. So it's in my interest to make sure that I or anyone of my people in the team are following the rules and guidelines. So if you don't have those guidelines and rules in place over here, then it's very easy to have the kind of problems that we have.
Recently in Japan, there was an incident where 3 manually disabled robots downloaded information from the internet and remade themselves and went on to kill people. What do you think the potential, the deathly potential of AI, and how do you think we can curb it better?
Well, I mean, we went through all of that, and again,
Even in many situations, it's been shown that they tend to break their own protocol and deem human beings to be inefficient.
right.
And most of these things are bypassed in this particular case.
So I don't know. The people who are designing that, the engineers in here need to design that system where either cannot be. So obviously, what you're saying is it's not a physical system, right? So would you say that those robots have gone mad, like people go mad or whatever it is? So we don't have a way.
The problem being is they're logically correct.
I don't know whose logic. Why are they not?
Human beings are inefficient. We are killing our time. It is logically correct.
And they're not. The robots are not killing the planet because they're not using any energy or what?
Everybody believes that human beings are the virus which is killing our own planet, and they're just doing what is logical.
I disagree. Again, logic is they're using a logic which is being fed to them, right? So we talk about algorithms are scopes, and so in their minds that's logical. You believe that that's logical, and that's your belief. And for other people, it may not be. It doesn't. So how do you kill those robots? I don't know. Maybe unplug them. Let's make some sense, right? So we start talking. And that's the problem, right? So even with the cars that are now computerized, they can be hacked. You don't need anybody to go and fly a bomb for themselves. You can just use a car as a lethal weapon. Is that right? That's an inherent problem that's going to happen. So we have to decide as humans that I think there's a bell-shaped curve for everything. And at some point, even that technology is, like I said, peaked, and after that, it's harmful. So that's why we need policies and all the rules in place. And of course,
we'll have a break.
Oh,
I have a query.
yeah.
When you talked about logo, is it the mindset of the child who was drawing it, or what is it actually?
No, it's a computer-generated image. It's not a child's imagination. So this woman, Stephanie, she was using the super composite program, and she just typed the words, and she was giving it a negative command. So it was asking it to produce images
Actually,
of,
it seems like something like that. It is like a
say, gave the word radio, what's
the
the
thing you
opposite
type a
of
description,
radio?
you type it creates
So in this case, she gave the command of logo, and this is what it is. So it's a creation of that super composite program. The work
post it on social media.
It's not the mindset of the women who was doing it.
no, but it's collecting all that information from the net, right? So that's what I'm saying. You could be viewing whatever you're viewing, and I peek over, and you know I'm going to peek over, so you're going to switch to something. But who knows what you were doing in the back? But you can't hide that from the whoever's collecting that. Can I hide nothing from Google?
So it takes up from everyone, it takes up, people takes up what are the queries and tries to build its own.
It's like you can hide whatever you're feeling from the world, but you can't hide it from your own self, right?
Okay, thanks, Dr. Aditi, for a very valuable lecture. Started with the basics and also touched upon on the importance of the data collection, and also educated now, like we now have to doubt or try to start questioning the data that has been collected, and also try to be self-critic, and also talked about the application part, trying to emphasize on the healthcare issues and also the ethics involved in the AI. Thanks a lot, and I request Vishal to hand over the memento as a token of gratitude to Dr. Aditi. We take a break for 10 minutes for tea and come back. Thank you. And the next session is by Dr. Shushma. the sapling as a token of appreciation. And I take the freedom to call Dr. Shushma as Dr. Spice Variant because she has been extensively working on the regulation of alternate splicing in cancer and aging. I welcome you, ma'am.
A warm welcome to all of you. I hope you all have enjoyed morning session. Now we have one more energetic speaker to us, Dr. Sushma Nagaraj. It's a great privilege to introduce Dr. Sushma. She is a professional computational biology unit, University of Bergen, Norway, and she is head of Node Elixir Norway. She is a professor Durham University, UK. She is the project leader of Elixir Norway and co-head of the Norwegian Elixir Node. Her research interests include transcriptomics, especially for alternative splicing regulation in aging, cancer, retinitis, and pigmentosa, and more recently, the biophysics of condensates. She is the group leader at the Computational Biology Unit and at the Department of Biosciences at the University of Bergen. The lab is interested in RNA biology, especially alternative splicing and aging, using multidisciplinary approaches, skilled in genomics, molecular biology, biochemistry, bioinformatics, and genetics. Her PhD focused in RNA biochemistry from University of Cambridge, postdoctoral training at University of Genetics, Institute of Genetics, Germany, Newcastle University, UK, and EMBL EBI, UK. She has received several honors and awards for her credit. STFS, she has received STFS Fellow EMBL EBI 2012-13. She is a postdoctoral researcher with a short biodata. We all welcome you, ma'am. Over to you.
Thank you for the very kind introduction, and thank you also for inviting me here. This is my second time in this seminar room, and it's really, really nice to be back again. Okay, so that's me. You now know. I was very curious as to what would come up because I didn't have a chance to look at. I hadn't given you this information, so you've clearly done the real, I think, and found talk. So that was great. Okay, so let's see. So I really want to thank you first of all for asking me here. I had a wonderful time earlier in the week when I came to visit. That was really nice. Thanks for hosting me. You're all, I guess, really in the room. It's been very nice. And the others there, I'm trying to look for. Yeah, there it is. So the other group of people I would like to thank straight away are the members of my group. I don't actually do anything, actually. I can't do anything anymore, really. They are the experts. So if I talk about work, it's their work I'm talking about. It's their research that I'm just presenting and then, I'm guiding them. However, it's theirs, and that's important to recognize. So they're a group of very talented PhD students and postdocs, and some little ones who we recruit very early in the process. This is a tough world, so we start very, very early. We are a very social lab, so we tend to have a lot of fun, including with our collaborators, so it's really wonderful to be here. Right, so very brief bio of me, just to give you an idea of what my journey in science has been. I started here in Bangalore with a BSc in chemistry and biology from National College in Basangudi. I then went on to do a master's in JNU and did a project, the master's project at NCBS in Bangalore. I then went to Cambridge for a PhD and was a department of biochemistry. Can you hear me okay? Okay. So this is the point at which I really began to look into bioinformatics. Because what was happening is I joined an RNA lab interested in three aspects of RNA biology. So this was alternative splicing, translation, and RNA localization. And the three PIs running RNA lab together, and so we had this very, not really interdisciplinary, but generally environment than usual, perhaps. This was a period where the human genome sequencing was also coming out, and I was quite excited that we have now all these DNA sequences and EST sequences, as they were called, express sequence tags, so RNA sequences. And I could begin to try to put them together and see, okay, how does the genome relate to the transcriptome? And to do that, I had to start to code. And that was the first time, really, that I started to pick up some coding on my own, and then went to proper lessons, and that was the beginning of my bioinformatics journey. I then went to did my PhD on alternative splicing. And thank you for the name that I've got. Another name I'm known by is actually Splice Girl, which is quite funny as well. That's not the other handle I've been given. So I went to the Institute of Genetics in Cologne because they were working on a genetic disease where the basis of the disease was missplicing. Okay, so I was, having done a very kind of, theoretical PhD, working very mechanistic, I wanted to really see, okay, is this stuff that I've done of any use? Are there any real people who care about this stuff? And so I went to a hospital environment to see if I could make a difference. And of course, as very often as you all can be when you're young, I was naive and I thought, okay, fine, I'm here now, it's going to be good, right? And you realize that over a period of a year or two, you make a tiny fraction of progress. But it's important that that progress is done properly, that's done thoroughly, and that takes time, and it's important to take that time. So that was that. I then moved to another genetics institute, finding this really interesting, that I could use the molecular biology and bioinformatics that I had learned to put it to use in a system and to understand mechanistic basis of disease, but also to contribute to the diagnostic pipelines that they were coming up with mutations that were unclassified. So we wanted to see perhaps splicing had a role to play in some of this unclassified variants. So that was something else that I was doing then. I then decided that I needed to spend some quality time doing bioinformatics only, and then I went to EBI, which is purely dry labs at the time. That was very good. And around this period, I also applied for independent positions, and shortly afterwards started my lab in Durham. A few years ago, 3.5 years ago, I moved my lab to the University of Bergen in Norway, which is where I am now. So that's a brief kind of travel. And in Norway, I do two things. One is to run my lab, and our interest is regulation gene expression at the RNA processing level. So we are not very much bothered about which disease we're looking at and so on. Of course, we are for the project, but in the larger mission of the lab kind of idea, we're interested in just finding out where is this RNA going from the point it's being made. From the point of transcription, where is it? We are often counting RNA when we look at RNA-seq. We don't often take into account, is it getting translated? Is it getting transported to the right place? Is it stuck in the nucleus? Is it there? Is it degraded? What is the rate of that? So we're very interested in asking those kind of questions and to look at regulation of gene expression from that kind of angle. We're often working in a use case, and this could be aging, it could be breast cancer, it could be retinitis pigmentosa. I'll tell you a little bit about that in a while. And we are often combining both the laboratory side of things and the computational biology. The other role I've taken on more recently is a role at Elixir. And Elixir is, I'll tell you more in the second part of the talk. What Elixir does is to coordinate life science data resources from across Europe. So they form one single infrastructure, so it makes it easier for researchers to find the kind of data they want, makes it easier for them to deposit their own results in a format that other people can use. This business doesn't bring papers. It brings me a lot of work. It's very rewarding because it's something that will outlast me, and it will stay there, and it's my current contribution to this research environment. And it's a very exciting space because in addition to just the science, I'm also getting a little bit more involved in policy and how actually science is being used, what are the issues facing those who are both using data and depositing data, and we understand both the importance of quality quality and the value of the data that people are making. Because in singular, perhaps the data is small, but put together, the data can be very valuable, and we must remember that most of the data we are dealing with has been generated using public money. So this is actually, people have paid for it, so you paid for it. So it's important that it is kept properly and that it is a resource that is also available to everyone. So that is something, it's a new thing that I've sort of started to do. But take a proper step back now, okay, and get into the science of it. So you all know we're made of 100 trillion cells and thereabouts, and we all know what a human cell looks like. What's very interesting is if you think about the cell and the DNA in it, and this can be sequenced, and you know all this, you've seen this with me yesterday especially, and you know it from before. So one might argue, okay, that the genome is like a program, it's got thousands of lines of code, it's got instructions, all that is fine. But something interesting that happens is that every cell in your body has that same program, right? And yet you're not going to confuse a neuron cell from, say, a skin cell. It's obviously very different. So there is clearly, this is not all the information that is needed. There are other things and other processes that govern what the cell ends up looking like. And this was one of the big findings, really, of the Human Genome Project, that we simply don't have enough genes. They found about 30,000 genes, and yet by then it was well known that there were, more than 100,000 proteins. So the point was, where are all these extra ones coming from? Where are all these extra proteins coming from? And this was this period of thought was, as you can see, around 2005, science was writing about it, and it was becoming increasingly clear from when the human genome sequence data started to be released at the turn of the century that this was going to be well, a mystery. So many of you will have seen this. I put this up simply to remind you to read this paper, the small opinion article, if you haven't done so already. It's just quite interesting. What I like about this is it's got a lot of opinion, a lot of thought. And we've stopped writing our thoughts down as scientists, as even professors. That's what the word means, right? That one professors. So one thinks and one talks about it. You say what you think about the world and then write about it and try to change the world, or at least, convince someone else about what you think. Or be convinced yourself that perhaps you were mistaken. This is our business, and yet we stop doing that quite often. We're now just writing results in papers. We write down and say, this is the result I got. What does it mean? Yes, discussion paragraph. What will I do in the future? Another discussion paragraph. Do we really write what we're thinking? There's very, very few fora for it anymore. And so I like this paper to see, well, just go and read a paper about how you should express your thoughts. And actually, next time you write a paper, try it. Of course, present your data, but write on what your opinion is, write on what the community's opinion is, and see how that will go. They will probably edit it out, but give it a go, right? And it'll be a good exercise just for yourself, even if it doesn't make it to your final paper. Anyway, the idea of the central dogma was, all of this stuff, you can read that. And there have been, obviously, modifications to it as it's come up over the years. And a couple of modifications, which is that genes don't always encode protein-coding RNAs. We now know there are lots and lots of non-coding RNAs. That's well established. The other thing is that eukaryotic genes are split. So what the central dogma said is, the DNA gives RNA gives protein, protein cannot go back, and the RNA is the mirror of the DNA, and we know that this is not necessarily the case. So what the concept I wanted to bring to you is that there are sequences that are exons. You can see them here in orange, and the intervening sequence are called introns. And many of you would have probably heard about it, but I'm aware that there are many first-year undergraduates in the audience, so I'm repeating some things that might be really blindingly obvious to many others here. So, to try and remember it, introns are basically interruptions in a coding sequence, okay? And what the cell needs to do is get rid of all these interrupting sequences, join all the coding sequences together, and then make the protein. So, and which is what is shown here. So you have transcription, you have capping, splicing, which is removing of introns, and polyadenylation. Mature RNA is then moved out from the nucleus of cytoplasm, undergoes translation, finally captured. So you might ask, what is the point then? Why bother with all these interruption sequences? Why not just have the coding sequences, right? So what the interrupting sequences gives you. Oh, I know what's happening. It's very important. Okay. So here it is. You can have just remove all the introns, you have that. But what you get by allowing some exons to be selected and some not to be selected is from one RNA, you can now make, I'll make a mistake here, there should be 5. But from one pre-mRNA, as it's called, you can end up a whole different bunch, whole different variation of mature mRNAs, right? You can start with the exact copy of the gene, of the DNA, and now by selecting some exons and shuffling them around, you can actually end up with more than one protein from one gene. And that's really what alternative splicing can do for the genome, if you like, if anyone is doing anything for anyone here, is this huge increase in diversity. And as many of you will know, that when films are made, for example, that's exactly what they do. They shoot all the bits here and there, and then splice them together. It's kind of the same thing, or the same concept anyway. So this leads to at least 3 times expansion. So you start with 20K human genes, you end up with over 1900K. So there's a huge expansion through alternative RNA splicing. It's important to remember it's not only splicing that gives rise to this expansion. There can be alternative polyadenylation, there can be alternative promoters. It's important to think critically about all the ways by which you can get variation, all the things that can happen once a gene is activated to the RNA being made, which RNAs are made, where do they go, how long do they survive. This is important to bear in mind. Okay, so this expanded protein diversity can have some very impressive examples. Okay, so one is a very popular example called DSCAN in Drosophila. And this gene generates 38,000 isoforms. And that number of isoforms from this one gene is about 3 times the total number of genes in Drosophila. So that's the extent to which you can get amplification of information that is stored inside one system. And this, of course, can have very interesting implications for those who are trying to store information using DNA as a storage molecule, right? So if you can actually put in splicing into the information storage systems, you could amplify that too. And so there have been articles written on this from a computer science perspective that this could be a cool thing to do. All right. So there needs to be some sequence signals for splicing. So there's a long chain of RNA, and you've got AUGCs, and there are some signals that show where an exon begins, where an exon ends. And you can see that here. So if you have a complete mRNA, we can just go to this one. Okay, so if you have a complete mRNA, and you can see that there is a start. Here is that's the wrong one. Okay, so then you have the complete mRNA, you have UTRs, you have one of the exons has the ATG start codon, and somewhere down there is a stop, and then there's some more untranslated region. Fine. And now there are some sequences that are kind of commonly seen, GTAG, and you don't need me to tell you that that is not enough information. To just have GT and AG is not enough information. This one's not good. Okay, now, so the way this signal is recognized is that various proteins, called lysosomal proteins, they go and bind at these boundary sequences, recognize, form what is called a large spliceosome, and the spliceosome then allows the RNA to be cut and pasted. So, there's clearly not enough information on sequence alone at these edges. So more a specification of what is going to be selected and not is given by other proteins called enhancers and silencers. So enhancers help these guys bind in the border, and silencers prevent them from binding in the border. And this basically decides which of the units or which of the pieces of RNA are kept and which ones are removed, discarded. The ones that are kept, we now know them as exons, and the ones that are removed, we know them as introns. And I deliberately say it this way because there is what we would call the exon, right? So if you draw a gene out and say, here's exon, here's intron, here's exon, here's intron, it's not us telling the cell that here's the exon. You define the property that something is an exon because it got selected. And what was removed is automatically an intron. And this is mobile. It can change in disease states, it can change through development. It's important to bear that in mind. I'll just keep an eye on it. So it's trying to connect to the internet, which it can't, and so it's so why study splicing regulation? I think you already mentioned this a little bit. I was interested in this because it's dysregulated in many diseases. So we wanted to see, okay, what happens? How can we study this process? And this regulation can be due to mutations. It could be in splicing factors. It could be in sequences, and it could be in the splice sites itself. So in diagnostic practice, they have been using a lot of programs, and last time I was here, I went through some of the programs with you, but they're quite common now, and I won't do that today. So the programs that can predict, not using AI or anything, really, lookup table programs, very, limited. They do what they do, and they do it well, which is to say that, okay, this particular mutation is associated with, say, a loss in exon inclusion. So if you have that mutation, then you should expect that that exon is gone. And if that exon is gone, we're not talking about a point mutation or a frame shift. What we're talking about is a whole chunk of sequence missing. And you need to then model the implications of that. So that kind of application in the clinic through the use of software that integrates this information is already in use. And today I want to give you an example of one disease that we are really working on, which has been a bit of a mystery, and we've already started to use some AI machine learning type approach. Again, I won't go into the details of the machine learning because you guys know quite a lot about it. You studied various examples of it, so I won't go into technical details of the use of this program or that, or this approach or different modeling system. But what I want you to take away from this is when you have a problem, how can you go about thinking about the disease and approaching it logically and saying, okay, are we going to see something here? So many of the techniques that you'll see here are not anything to be very excited about. You know about them. But we'll go instead through the logic of what and why we did what we did. So the retinitis pigmentosa, we were interested in it and we came to work on it because mutations in splicing factors cause retinitis pigmentosa. And it's a leading cause of blindness. The most interesting thing for us was all the above genes are splicing factors. They're expressed in every single cell. They're required for normal cellular function. And yet, the patients are born with perfect 20/20 vision, and in their 20s will start to go blind. This was a bit of a mystery. Why does a splicing factor mutation end up causing blindness? What is it actually affecting? How can we begin to study this? So we went to what's called patient-derived model system in collaboration with the Nicola, who carried out all of the stem cell work. And I'll explain this very briefly. So when patients come into clinic, you can take a small patch of skin called a punch sample. So it's a little thin dot of skin. And from that, when you put that in culture, you can get patient fibroblast cells. So now you've got the patient genome there, right? So you can do that. You can now turn those cells, fibroblasts, into induced pluripotent stem cells, called iPSC cells. this is the so-called Yamanaka method. This is now very well established. So there are 4 genes that are transfected into these cells that turn those fibroblast cells into stem cells. Now what you can do is you can take these stem cells and make whatever it is you want from it. You can make brain cells, you can make eye cells, gut cells, liver cells, whatever. So what was done in this project is that we created retinal pigmented epithelial cells and created retinal organoids, which are photoreceptors. So these are now model systems of the patient. They have the patient genome, and this was done multiple times for a whole lot of patients. So this is very laborious. It takes 2 years to get from here to here, just in time for the iPSC cells to actually develop into RPE cells. It can take up to 2 years. So this is very laborious, and so the large number is also a good question to ask. Was only about 9 who actually made it all the way through. We started with 15 patients. Bear in mind, this is also a rare disease. So the incidence of this is not that high. So we have a patient group with retinal pigmented epithelial cells, and we have the same patient. We have 3D with retinal organoids, photoreceptor cells. Okay, now can we do something with this? Bearing in mind that, okay, this is not perfect, right? What you ideally want is you want 200, 500 patients, you want them from across the world, you want to have their actual retina and their actual thing. You can't have that, right? You can't take retina from anyone. You can't have a piece of the liver kidney. So this is kind of where we were at that point. And I'll tell you at the end of the talk where we are now. So we got the photoreceptor groups and so on, and got RNA-seq data from them, among other readouts. So I'll tell you just about the RNA-seq just now. So okay, that's good. They're all nicely separated. You want to see that, and you also want to see that there's some close clustering by tissue between patients as well. So that was nice. Okay. As a surprise for those who were not doing the splicing at the time, there were hardly any genes actually that were different between patient and control. If you just standard differential gene expression, if you just count which RNAs are differentially expressed in control versus patient, a handful of them. And when you look at this lot, they are mostly sort of stress-related genes. They are degeneration genes, genes involved in cell polarity. And you don't really see very much. There were very, very few genes. But when you look at, if you try to look at what are the alternatively spliced isoforms for the same genes, is it possible that there are isoforms being produced that are different between control and patient? You see quite a bit. And differential splicing, when we look at what types of genes they are, we see cilium assembly, we saw all this kind of stuff, right? So what we did see is that ciliary genes are misspliced in retinal cells. And what does cilia have to do with eyes? And what is it that we are seeing here? We also see that splicing itself is splicing genes are being misspliced. So we think that there's a big amplification of the effect here. Okay, so one thing that we could then go and do is look at the cilia. And if you look at control retinal pigmented epithelial cells make very nice sharp cilia. And cilia are tiny appendages that every cell has. And they're believed to be hubs of signaling. And their role or their function in cells is being worked out. And there are many new functions coming up all the time. But indeed, just as our bioinformatics had shown us, we could see that the cilia were really, really messed up in patients. So this was fine. It was good to see, of course. And what we could now do is that if we CRISPR corrected it, so if we genetically engineered the patient cells to become control, you got the cilia back. In all of the 9 cases, in all of the replicates of the 9 patients. So this is very good. So what we are now, that's published, you can read more about it if you like. And I just think I'll just finish with this part of the talk here. So it looked like we solved the mystery of why the eye is affected, because the eye is a modified cilia at the end. The photoreceptor cells, especially, are modified cilia, stacked and filled with rhodopsin. And so that makes sense that the ciliary genes are affected. The mystery we have not solved is you start with a mutation in splicing factors. Why are you getting, what are the genes that can be affected when the splicing factor goes well out of balance? As I said, so can we actually predict which genes are likely to be affected, bearing in mind that every patient is different? We need to integrate how much spliceosome components they are making to begin with. We need to integrate how much enhancers and silencers they are producing, and what the sequence is. So this is quite a large puzzle to try to integrate all of these things. And to do that would give us what we might want to call the splicing code, right? So if you have a certain code and there's a break in it, you can then predict where you're going, what you're getting. But we actually don't know what this code is. We can measure, we have access to measure it, but we don't really know how this works. And in many ways, this is quite a nice problem. It is quite a nice environment in which you can apply such a apply machine learning type approaches. And to do that, I've kind of just put down a few things that you need before you go to apply machine learning to such complex problems. One is, for example, you need the molecular biology to be established. So you can map all the steps in the splicing reactions. You know what you're expecting to get. You should know that what you're measuring has high fidelity. So what we've done in our RNA-seq experiments is that we spike them with some known isoforms, and then we measure what we get back so that we reduce the noise coming from there. So the data going into the system is robust enough so we can actually say something about how the algorithms are performing when we get the output back. Otherwise, if we blindly trust the output we're getting back, it could be, if you like, the algorithm's imagination, which we now know is quite robust, right? The algorithm is pretty good at making stuff up. So that's something. We need, of course, the genomics to understand the whole systemic role of gamma. That's fine. The genetics to work out the function of isoforms and their importance, evolutionary importance in general, but in our particular example, it doesn't matter it doesn't matter which combination of the SNPs are working together, or it doesn't matter or do they work the same in isolation. And to do all of this stuff here, we need this iPSC system that we have, where you have a lot of induced pluripotent stem cells. You have the matching CRISPR-corrected control, the case in which the patient is no longer a patient. What if that mutation was never there? How would the gene expression profile look like? And just even though we only have now, at that point we only had 9, they're now about 30, those are still very small numbers, but that's kind of all we're going to get. But we can make more by taking other genotypes and inducing mutations now in hypothetical patients, right? Because there is no person with that mutation, but you can take any control cell line, put in the splicing factor mutations, and then run through these steps to generate high-quality data from which you can actually derive conclusions. And for that, of course, we need computational biologists. It's only when you've got all of this stuff in place that you can really go to the step to start the modeling. Because otherwise, you're going to just model something, you're going to model hypothetical inputs and outputs, and it's not going to make any sense. And you need the controls to say you need to have error measures in place. You need to be able to measure it, you need to quantify that, and put that into the models. Otherwise, we will just produce another one paper, probably in a good journal, that says, okay, we've also discovered the splicing code. If you Google splicing code, you'll find about 4 or 5 papers that will allegedly cracked it. And yet here we are talking about, we still don't know, we can't predict it. If you know the code, then you should be able to predict it, and yet there are no good predictors for splicing outcome given switches and splicing factors. And there are some methods, but these methods do not claim to have a code. They do not claim to discover the splicing code. These are tools to carry out operations. Okay, so I'm going to leave that there and perhaps tell you about the results of this pumpkin next, right? Oh, I don't know what it is. But very briefly, I'm going to tell you about another project in lab. How much time do I have? 20? Great, okay, thank you. I'll take less than that. So, this is a student, postdoc in my group, who's very interested in, I think I skipped a slide. Yeah. So we have a project in the group looking at breast cancer patient stratification based on the reaction to various drugs and the reaction in terms of the signaling pathways. So we're not looking at patient outcome, we actually have a cell line and various cell lines, also from patients, and we ask if you give drug ABC to different genotypes in different patients, how does the signaling pathway respond? And so this is a signaling modeling project. And one of the practical problems that we needed to solve here is that if we take tumor biopsies that we receive from the hospital, these are needle biopsies, and we just get what we get. And sometimes you might have 20% tumor remaining control, you might have different types of cells, or sometimes you may have 80% tumor, and yet you're going to try and attribute and say, okay, this patient has more signaling of something, right? Or less signaling signature in some other signaling pathway. So we need to be able to deconvolve and say tumors are not like, this is tumor, this is control. They're not homogeneous things, they're very heterogeneous populations of cells. And what Francisca is doing is trying to deconvolute this data to come out and say, we've got that percentage of this percent of that, and so on. So that's, in doing that, she's developed a machine learning based approach that she called DTD, and she finds that DTD performs very well against some well-known, commonly used systems such as Cyberspace. So this is a quick plug for Francisca's thing, which is quite nice, and it solves for us using machine learning a very practical problem that we need to know what percentage of tissue versus control cells can be attributed for a given biopsy so that the clinical guys can interpret what the models are saying. So, and move forward. So that in this project, Mesistrat, this is a stratification of breast cancer, I wanted to highlight something on this site. So another role that my lab plays, in addition to helping them with bioinformatics, so we do a lot of this, helping people with their bioinformatics. In addition, we have the role of so-called data management platform. So what is this data management platform? Mesistrat is a very big project. It's got about 40 project members, and it's over 11 universities across Europe. So there are lots of people involved. There's a lot of potential for massive amounts of confusion if we send stuff by email to each other, or if you just have Zoom meetings and say, okay, fine, I'll email you later, or I'll put it on the drive, it's not going to work. And we know that for quite a while that this kind of thing doesn't work. It doesn't even work inside your own group once people go away and once people finish their PhDs or so. And it doesn't, I think, even work for an individual, because I don't remember where I put some email somewhere if I haven't cataloged it properly. Unless, of course, I can ask Google, there is that these days. But I still need to know the correct keyword. Much easier if you know exactly where to go and look for what type of data. And that's what's being done for a lot of bioinformatics across the world. And when we submitted the grant, we wrote this kind of made this figure, and we said, okay, we will put everything into this thing called BasiSeek. Seek is a platform for sharing and storing data during the project, and it's based on something called Fairden Hub. And Fairden Hub is a publicly available system that you can adopt to manage your research data well. You can put anything in it. You can put holiday photographs if you like, but it's a bit cumbersome if you use it for that. But what BasiSeek does, and what Fairden does, is creates a system by which if you have, say, metabolite data, it will make you put it in such a format at the beginning of the experiment, will make you put it in such a format that it can be pushed to a repository called Metabolites later. Metabolites is also a free to use, any of you can log into that and use it. And the same goes for sequencing data, it pushes it to EMA, protein interactive data takes it to intact. But at the time when a scientist puts their experiment in and says, I did this experiment in interactive data, it will make you formulate the tables in such a way that when you come to publishing, or maybe you don't publish a piece of data but just want to share it, you can push it to Intact. Intact then shares it out to the whole world. So anyone can log into Intact and ask, does my protein interact with some other protein? Oh, who does my protein interact with? So this is something that we were setting up. And following on from SEC was a very good time for me to join NXIR, which does this whole data management at scale across Europe. So we've done it for one project. We then thought, can we actually do more of this? And this is the kind of stuff you get, right? So many of you who are computational biologists or deal with people from the experimental side, or indeed from just any other field, also for biologists who deal with computational people, I can tell you now, and so also the guys in the lab are complaining, is that people just tell us, give us data, and they don't tell us what is what properly. And they say it's all labeled, you don't do it. And it's not that easy, you can't do that. You need systems to be well organized. And this, how should you share data? In a fair way. And what does fair stand for? It should be findable. And it should be human, or preferably machine readable, attached to persistent identifiers. So anyone should be able to find it. It should be accessible, and again can be found and retrieved. It's all fine to find it, right? You also need to be able to get it. Then it needs to be interoperable. It means you can exchange, use it between systems, to perform functions. And it needs to be reusable. So it means that you need to be able to reuse that data for some other analysis, for a different meta-analysis, to combine different types of data and do something. So these are important to do. And then this was very nice, because this is exactly what these systems are. They're all fair data systems and allow you to run such computers. And so the reason I bring this here is that in order to perform machine learning, your data needs to be like that. It's to get enough, to be able to collect enough data to be able to run these kind of things. You can't do that unless you yourself have produced big enough data that can be used for machine learning. If you want to integrate across the world and do this, in an ideal environment, you don't want to go and put data into the correct format every time. We want it to be in such a way that we can employ robots to do something useful for us now, to crawl the web, collect the data from these kind of formats, in these kind of formats. And what I can do is I can send a bot into that and let it run my program. And it can only do that if it is in a known format that is fair. So that is the effort we are very much involved in. And there are, to make your data fair, there are lots of methods, there's lots of tutorials available, or formats available, and these formats are standardized, but broadly, you should use standard formats. So if you save your file, don't save them in some kind of funky language you just invented day before yesterday afternoon, right? So let's put it in the standard format. What is a standard format? A standard format is whatever your intended community generally uses. So then your data is findable, it's reusable, and all of that stuff. Supply metadata. If you don't know what the complete metadata is, who is the patient, what conditions, when, where, how, all of this is useful information for the person or people who are going to use your data later. It may be useful information for you in 2 years' time because you've forgotten what you did, right? So it's very important to do that. And again, similarly, there's a bunch of things. Put it in the stable repository like the ones I showed you, and then also cite other people's tools, share your data, and also encourage others to do this, because by working together, we massively amplify our own capabilities. Because now instead of 1 or 2 data sets, we have loads of them, and they are interoperable. And this is exactly what Elixir does. So Elixir is, its mission, if you like, is to operate a sustainable European infrastructure, supporting life science research and its translation to society, bioindustries, environment, and medicine. So we handle data, we help people make it fair, and set up systems. So this is like infrastructure. It's like making roadways for data. It can come across as very boring. But actually, it's data mobilization is an increasing area that needs to be developed, for, especially for life sciences at the moment. So there are many, many job opportunities for those of you who understand the value of data and how you can actually handle it and make it more available and more shared. So to make it available and shared doesn't mean just give it to everybody. That isn't the point. The point is that you need to know how it should be given and to whom it should be given and when. So this is all the member countries of Elixir. So every country has various members, and this is our current activity in Norway under Elixir 3, which is the project I'm heading. And I want to give you just one example, is that often end-user requests will be, give me a workbench, I need data management, it needs to be easy to use, secure, reproduce, integrated, blah, blah. This is what a lab person would ask for. This is what it ends up inside Elixir. And it's a service that people can then go and use. So they would have a system where they can compute, they can store, they can compute, they can export the analysis, and eventually push it to the world. And there are ways to use it using Galaxy from UCSC, UCSF. The login in the Norwegian context only, which makes sense, is something called FIDE, which means that every Norwegian student and or member of the university will typically have the so-called FIDE login. Okay, so you have a common login that reduces the barrier to go and use the system. But I can also invite anyone I want as a collaborator, so there's also a non-FIDE login. But this is just infrastructure, right? But infrastructure is never just infrastructure. We take it for granted that we have all of these things around us, but you realize it when it's not there. And you can also see when it is there, how much faster you can move and how much more you can achieve when such infrastructure have been built and are built with the users in mind. So the user interaction is important. We want to know what people want and then build it, and not just build something and give it to them to use, because then they might find something else. So this whole user thing is very important. All of that was for non-sensitive data. And what also it does is to help with hosting and rubber stamping certain research services. One of them that you might have heard of is ChopChop, which is used to generate CRISPR guides. That's one that's made in my department. But there are many others across the space where Elixir has been involved. We have a lot of users, including, as you can see, users here in India. So there are users all over the world, and this is important because this is public data. This is not our private. We're making it available and easy, but it's not ours. And then Elixir Norway has a help desk which does various kinds of services. It's jointly operated across 5 universities with a ticketing system. This is again a way to serve the research community and say, do you have a question as an undergraduate, as a postgrad? Not questions where you have to write an essay and we write it for you. That doesn't happen. That doesn't work. But are you stuck with a research problem? Do you want to come to us with the data? There'll be someone there who will eventually, as in within 3 days, pick up your email and talk to you about it. But there's one point that is worth mentioning is a controlled access to data sharing. So you might not always want to put all types of data out to the public and say, okay guys, take it, go. And not all data is suitable for this. There's sensitive data such as human sequence data. We don't want our human sequence data to just be anywhere. So that needs to be shared in a secure manner, in a sensitive manner. And again, Elixir has been working together with all our European counterparts in creating systems where data can be stored securely and operations can be performed on the data. So the data doesn't move. Data stays where it's produced, but the programs come and go. So you send programs in what are called containers, and the containers and these programs can be anything. So again, if you want to do machine learning type work, you will have to containerize a machine learning program. It comes to each country, picks up the information it needs, and moves on. And so this is how we will need to do these kind of research. And so I think that's just to scheme that too. Right, so this is something I just said, that the data existence description is made public, but not the data itself. So you know that if you want to do a certain operation, you need to go to Spain, because that's the data they have. You, meaning the bot, needs to go to Spain. Run the analysis, give you the data back. The next stop might be another country, but now these are countries, so different countries with different legislations. They all need to agree to this. How are we going to do it? There are lots of laws around this. We need to understand those laws, and we need to operate within the laws. This is of obvious importance. All of this requires approval and signed agreements. And it needs to be quick. As a researcher, you cannot sit and wait for 2 years before your agreements are signed, because the question may be of no relevance at that point. So what Elixir does is it facilitates all of these by saying we know the agreements coming up in France, we know the agreements coming up in Spain versus Norway, or the UK, or India for that matter, because we want to move data in between. And we know we cannot move data between any of these countries. So we want to move knowledge between, or we want to move analysis between spaces. And the systems need to be prepared in advance so that when the researcher shows up, they actually experience a wonderful, nice, hopefully smooth transition to doing the experiments. And much of this was pushed upon us. We didn't want to do any of this, right? As researchers, we were very happy to sharing data nicely, behaving nicely, and all this. But there's a legal basis to all this through GDPR, which just basically says you can't move any data anywhere. You can't have sensitive data. And as a scientist, 3 years ago, I was simply told no more sequence data analysis on your own servers. Just no. And so what are we supposed to do? We need to put in a secure server. What about the data we're generating with partners in the UK, because we're looking at rare disease? We can't do it. This was a huge, huge barrier, and much of that has been overcome now through these kind of systems. I should be done. I'm literally in the last. So now in the beginning stages of this, and just in September, this was published. The tool to do this federated data analysis across these countries. Not every country is in there yet, but some of these countries where you can see these colored boxes showing up. The others are in development. And so we have basically this, that you have an internet exposed part, and then you have this closed area wherein you can run database programs and similar such secure environment programs. And it's in this environment that we will have to carry out all the kinds of analysis we want to do. And just to give, this will be the environment in which we will do personalized medicine and health. So if you're thinking of algorithms, if you're thinking of approaches that take personalized medicine as your end goal, you have to think about data security. And how are you going to do this? Where are you actually going to put it? Do you have the required legal compliance statements? And what will be the individual steps in the infrastructure? We have to think about it. If you're not ready for this, we can do the science on personalized medicine already in individual cases, in small numbers of cases. But if we want a place where everybody will have an electronic health record, where we would know that we would have personalized monitors on our arms, as we are told, right? This is the future. That we would all have monitors that would be reading our blood pressure, blah blah blah, and we would get a formulated pill that says, okay, today take this, tomorrow take something else, and the after tomorrow, or so on. To get to that point, we will not be able to do that by keeping the data on somewhere, some server somewhere. This will be allowed and should be. So this is the environment where we will have to work, and there are increased preparations both in Europe and also in the US in this direction with NIH. And I'm less aware of what the structure of thinking in this area is in India, but I'd be very interested to explore this further. And it's something that I think we want to take forward from these meetings. Okay, so this is something I said already. Yeah, but this is now just a rough thing of what does Elixir do. It does standards, looks at interfaces, secure federated clouds, provides tools and services. So it's really very, and all of this is free, right? So any of you can come and use Elixir services. They're free to you all. A reminder on fair data, what it means. And finally, this is Elixir Norway group. This is all the people across Norway who work on the Elixir project. And that's it from me. Thank you very much.
Thanks, Dr. Sushma, for the informative talk. The session is open for questions. Students, please.
So yeah, in that disease related to blindness, it's called retinitis pigmentosa. So the first step what you do is you take the fibroblasts of the skin, right? Like any random part of the skin, right? You take. Yeah. So my question is this: how is that random part of the skin, like how are you able to obtain the retinal epithelial cell? Because that's nowhere related to the eye. It's a part of the skin, right? So how do you get the eye from there?
Yeah. So you start with the skin punch.
Yeah.
You put that in cell culture. Okay. The fibroblasts come out.
Yeah.
Now you have fibroblasts that represent the genome of the person from whom you took it.
Okay.
Right?
Yeah.
And we take the skin only because it's a bit that people are happy to give.
Okay.
No one agrees when you ask them for a neuron, right? So the skin, they're happy to give it, and the skin multiplies easily in culture. So you take that. And now, but there is something called an iPSC technology. It's also called the Yamanaka method. And what was shown is that if you start with a somatic cell like this, you can convert a somatic cell into a stem cell by transfecting 4 factors into it. SOX3, nano, SOX4, Octfo, yes, SOX3, Octfo, Kela, and together we manage. So the 4 factors are transfected. These are stably transfected into cells, and when you let the cells recover from the transfections, those that have recovered, they actually revert back in developmental time and become stem cells. Again, stem cells have the property that you can, by giving them the right environment to grow, you can turn them into different things. So one of the first things that they were turned into were neuronal cells. Neuronal cells are relatively easy to make, actually. And but nowadays, people are able to make a whole lot of different types of cells. So these factors that we have, we've made them in also produce kidney cells. The reason being that kidney cells have cilia. The cilia are important, but they're clearly not eyes. So, but the patients actually, interestingly, don't get a strong kidney phenotype. So we want to know how we, on the right track here. And so we're in this phase of the program where we're comparing between kidney, retinal pigmentosa, and photoreceptor cells.
So like, are you trying to say that from one patch of skin cell, you can get the whole human body? Like the whole human?
In pieces.
Yeah, you can get
not
that.
make a human.
They only have some features. So it's
Yeah,
not
that's
like
right.
it's an actual, the skin cell that is going to be mimic some features of the eye cell or kidney cell, because there are still very many different cell types in the eye or the kidney or the blood. So it's only going to be reproducing or mimicking the features that are maybe like represented. So you can't really call it like a true eye cell or a kidney cell or a gut cell.
In any case, it's only a cell, right? Whereas all these are tissues. And the tissues, there are very few tissues in any case which are organs that are made of just one type of cell. You say. So even if we got to the stage where we made perfect cells that were 100% representative, you would still only have the cell. You would not have the whole tissue. And this is an area where people continue to try and make it more and more close to physiological systems.
In fact, you have the reverse problem if you take primary cells. So for example, if you take a neuron cell taken out of your cortex and you culture them, they will be different. So you also have the reverse problem where they actually lose their phenotype. Or for example, you take gas in our cells and culture them, they will become ductal cell types very quickly. So there are things that are keeping them in their phenotype in that context, and when you take them out of context, then they become something else. So that doesn't mean that they were, that's the inherent nature, right? So like Sushma said, that each of our cells still have the same genome, and it's kept in its place because of context. So it is interesting that they don't develop any other ciliary disease, or hearing loss, or other.
So there are sporadic patients who have hearing loss and all this. We also found out something that's sociological, right? So when you talk to the doctors who are really dealing with these patients, which is often in primary care, they tell you that, okay, you know, if someone is going blind, they're not particularly bothered if their hearing is a little bit worse, or if their kidney is a little bit not great, so their skin is not looking good. They don't really develop acute kidney issues, they do not develop acute hearing loss. But now that we've gone back and asked these questions, actually there are deficiencies there too. It's just that going blind is one of the first things that happens, and maybe the eye tissue is more sensitive. Because, and we have now more evidence for this, showing that the eye is heavily dependent on splicing, alternative splicing to maintain its differentiated states, whereas the kidney becomes less so. And the ratios of different splicing factors and so on are very tiny, have very tight margins in the eye. So when something goes wrong, it's not the eye that suffers first.
So I have one more doubt, like how did you use alternative splicing in the treatment of this disease, this blindness disease? I didn't get that.
We haven't used it yet, but we will.
Okay.
I didn't talk about it. So eye disease treatment is one of the systems in which really delivery of oligos has been quite successful. You can deliver it through eye drops, and what you can do is, so there's AAV, a viral mediated transfer of the corrected genes just into the eyes. And in trials for other eye diseases, this has worked very well. So there's currently a project ongoing with two of my collaborators who are closer to the patients to do exactly this. So they're designing an antisense to correct the PRP31 mutation, and you'll see what happens. That's what it is.
With this, we have come to the end of the session, and I thank Dr. Sushma, who started with the talk with the self-introduction, followed by, and I would really appreciate Dr. Sushma for simplifying the entire complicated process of alternate splicing. And she also talked about the data management in a fair manner, how it should be used in a fair manner, using Alexa. And the case studies she discussed was the misregulation of the alternate splicing, that is the retinitis pigmentosa. Thanks a lot, Dr. Sushma, for the wonderful talk, and also thank Dr. Aditi. Both the speakers were very, have given a very valuable talk, and also like it is an insight to, and we also, I hope everyone have gained insight on what is happening in the area of AI/ML, especially in healthcare. Definitely in the morning, Dr. Aditi gave it in a holistic manner. Thanks, and I request Spurti to present Dr. Sushma the memento as a token of gratitude. Thanks, Dr. Spurti. And we break for the lunch and come back again for the oral presentations. And the oral presentations will happen in the 4 seminar halls: Biotech Seminar Hall, Mechanical, MCA and IEM Seminar Hall. And I request students to, you please come here, we'll tell you where to go at 2 o'clock. Thank you.